This should be the very last lecture for Chapter 3, and I apologize for the length of the chapter. It is a long review chapter, but it does have some very important information, again, for the TEAS test and for preparing you for anatomy and physiology. So we went over the parts of the cell, including the cytoskeleton, microfilaments, and intermediate filaments, and now microtubules. And we went over some of the organelles, but now at the end of the slideshow, there's also some notes on each individual organelle or cell part. So it's a little bit more review, but anyway, we're going to go through this, and then we're going to talk about how the DNA, which is located inside the nucleus, has genes that code for proteins, and then that should be it for this chapter. But we ended up with microtubules are the largest of the cytoskeleton fibers. So we had the microfilaments, which include microfilaments called actin, which are important in movement, muscle contraction, and then we had intermediate filaments, and now microtubules. And the microtubules' role is going to be to anchor the organelles in place inside the cell, so provide a little bit of structure inside of the cell, and also to form the spindle during cell division. So the microtubules, we also call them spindle fibers because they attach to chromosomes and help pull the sister chromatids apart during cell division. Now, microvilli are areas of the plasma membrane that are folded. So they're small finger-shaped projections of the plasma membrane on some cells. And what they do is they increase the surface area of the membrane. So instead of having a flat, smooth membrane, you have all these folds, and these folds increase the surface area so that more nutrients can be absorbed, more oxygen can be absorbed into that cell. You know, increasing the surface area means more absorption can occur. Centrioles are the structures that also help in cell division. They produce the spindle fibers, which are microtubules, that pull the chromosomes apart during cell division. Then we have cilia, which the only place we're going to find cilia are in males, they will help to move the sperm along before the sperm are capable of swimming. In females, they can be found in the female reproductive tract as well. And then probably the most common place that we have cilia, little hairs that line the plasma membrane of a cell, is going to be lining our respiratory tract. And that's going to help to sweep mucus and trapped pathogens and debris and just stuff that can make you sick. If it gets deeper into your lungs, that cilia will help sweep all of that stuff out so that you can cough it out or cough it up. So cilia are like little hairs that line the plasma membrane, and flagella is a single long hair, and it functions in movement. And of course, in humans, we really just see the flagella in the sperm. Ribosomes manufacture proteins. There are ribosomes that are free in the cytoplasm and ribosomes that are attached to the endoplasmic reticulum. The endoplasmic reticulum that contains ribosomes is called the rough ER. Then we have proteasomes, which contain protein breaking or proteolytic enzymes, which we know as proteases. So they recycle proteins. We have the smooth and the rough ER, or endoplasmic reticulum. The rough ER is involved in synthesizing proteins, and the smooth ER is involved in synthesizing lipids and carbohydrates. I will go through. For the smooth ER, it says it has no ribosomes. It synthesizes lipids and carbohydrates, such as phospholipids that form the plasma membrane, cholesterol that's embedded within the phospholipid bilayer in the plasma membranes, Steroid hormones like cholesterol, and cholesterol is the precursor to testosterone and estrogen. Triglycerides or body fat, glycogen, which is the storage form of glucose in skeletal muscle and liver cells. Then we have the rough ER, which is involved in proteins, in producing proteins and packaging them. Golgi apparatus modifies and packages products of the endoplasmic reticulum. And it also forms vesicles. It packages enzymes within vesicles, and we call those vesicles lysosomes. Lysosomes are vesicles filled with digestive enzymes. They can remove damaged organelles. They can also break down damaged or diseased cells, so they can fight against disease. Peroxisomes also break down, but they contain enzymes that break down substances, but mostly it's fatty acids. They're more involved in breaking down fatty acids, and the reason they're called peroxisomes is because they have a byproduct that is toxic. It is hydrogen peroxide. So inside of the peroxisome, we also have to have an enzyme that will break that hydrogen peroxide down into water and oxygen, and that way it won't be toxic to us. So peroxisomes and lysosomes are breakdown vesicles, but peroxisomes are just mainly for fatty acids, whereas lysosomes are for, just about anything. Mitochondria is where the ATP is synthesized. This is where cellular respiration occurs. Okay, we have some checkpoint questions. Cells lining the small intestine have numerous finger-like projections on their free surface. What are these structures and what is their function? These are microvilli, and they function in increasing the surface area so the small intestine can absorb more nutrients. Absence of centrioles means a cell can't divide. Why certain cells in the ovaries and testes have large amounts of smooth ER? Well, that's because the ovaries produce estrogen and the testes produce testosterone, and those are both steroid hormones, which are lipids, and the smooth ER functions in producing lipids. Number 19, what does the presence of many mitochondria imply about a cell's energy requirements? It must have a high energy requirement. All right, the nucleus is the control center of the cell, and it does contain the DNA in the form of chromosomes. Inside the nucleus, we have the nucleoli, or nucleolus is singular, and what the nucleolus does is synthesize ribosomal RNA, which forms ribosomes. Chromosomes contain DNA and pieces of DNA called genes. Human body cells have 23 pairs of chromosomes, which of course that comes out to be 46 total. And chromatin is the loosely coiled DNA that is found in cells that are not dividing. So if a cell is not dividing, the DNA takes the form that we call chromatin. If the cell is actively dividing, then the DNA is in the form we call chromosomes. A gene is defined as a functional unit of heredity. It's a piece of DNA basically that codes for a protein. Gene codes for a specific protein. Now, so how does protein synthesis work? It occurs in two stages. The first stage is called transcription, which occurs in the nucleus, and translation occurs in the cytoplasm. Transcription is when DNA codes for messenger RNA. I meant for that to be I'll just forget this. All right. So transcription is when the DNA codes for messenger RNA. This happens in the nucleus, and that messenger RNA takes the DNA code to the cytoplasm. And translation occurs in the cytoplasm on a ribosome. The messenger RNA attaches to a ribosome. the messenger RNA contains codons, which are groups of 3 nucleotides, 3 DNA nucleo, 3 messenger RNA nucleotides. And each 3 nucleotides codes for a specific amino acid. So what happens is there is a molecule called transfer RNA that contains an anticodon that is complementary to the messenger RNA codon. Probably show you something. Okay, so let's say that, for example, you know that the DNA code consists of A's, T's, C's, and G's, all right, which is adenine, cytosine, thymine, and guanine. So in the DNA code, every 3 nucleotides, like here you see AAA, which is 3 adenines in a row, every 3 nucleotides is going to code for an amino acid eventually. But what's going to happen is the DNA can't leave the nucleus, so the DNA has to make a copy of the code in the form of messenger RNA. So the messenger RNA is going to be complementary to the DNA. So if the DNA has an A, then the complementary nucleotide is a U. If the DNA has a C, the complementary nucleotide is a G. If the DNA has a T, the complementary nucleotide is an A. And if the DNA has a G, the complementary nucleotide is a C. And the reason up here that you have a U complementary to A rather than T is because RNA only has U, or uracil, and DNA has thymine. So RNA has uracil in the place of thymine. So if the DNA has 3 A's, then what's complementary to those A's are 3 U's. The messenger RNA codon, which would be the code for a particular amino acid, would be UUU. Now the transfer RNA is going to contain an anticodon so that it will know which spot on the messenger RNA that it should match up with or pair up with. So it's also going to contain an amino acid. In this case, the amino acid is phenylalanine. The transfer RNA is going to have phenylalanine attached, and it's going to come over and match up with the messenger RNA codon UUU because the anticodon AAA is complementary to the UUU. Now let's just do one more example. Let's say that the DNA code is TAC. The DNA triplet code is TAC. So what's going to be complementary to T in the messenger RNA is A. Complementary to A is U. Complementary to C is G. AUG is the codon that we also call the start codon, and it always codes for methionine, which is the amino acid that you find at the first amino acid that you find in any protein. So the transfer RNA is going to have methionine attached to it, and it's going to know which spot on the messenger RNA to go to bring the methionine to, because it has an anticodon, UAC, that is complementary to the codon, to the messenger RNA codon. So A is complementary to U, U is complementary to A, G is complementary to C. So UAC is going to match up with AUG, and that's where the methionine is going to go. Then translation is where and that's what I just explained to you was translation. Translation occurs in the cytoplasm. Okay, so what you see is in the nucleus, the DNA codes for a molecule of messenger RNA. The messenger RNA travels out of the nucleus into the cytoplasm. It attaches to a ribosome, which is this blue structure. And so the messenger RNA has the codon AUG at the beginning. A transfer RNA containing amino acid 1 is going to be methionine. See how it says first amino acid, methionine. Transfer RNA carrying methionine is going to bring it to that correct spot on the messenger RNA because the anticodon on the tRNA is complementary to the codon on the mRNA. And so the methionine, or amino acid 1, this one here, is going to be the first one in to form the protein. And then translation just continues on until all of the amino acids that form the protein are, have been added.